Prostate cancer is the most commonly diagnosed form of cancer in Canadian men, with 1 in every 8 being diagnosed at some point in their life and 1 in every 27 dying from it. My research looks at the expression of different genes at different stages of prostate cancer progression. And there are 4 main stages to prostate cancer progression. The first 2 are known as benign, so in these cases the cancer is localized to the prostate and it is unlikely to cause any harm. Stages 3 and 4, however, are known as a malignant cancer, where the cancer leaves the prostate and spreads to other tissues, and this can lead to death if not treated properly. However, many of these cases are slow to progress, and many individuals will die from other complications before they would die of the prostate cancer. If prostate cancer is detected early enough, it is easily treatable. However, since the treatment is invasive and risky, it is essential to determine who is likely to progress from the benign cancer to the malignant cancer, so we can determine who would need the treatment and who would not. My project is done in collaboration with the computer science department, who use computer algorithms on clinical databases to detect novel splice variants of different genes, which are commonly seen in different stages of prostate cancer progression. These alternative gene isoforms will often lead to different protein functions and structures, which will alter cellular function and can have an effect on cancer. One of the isoforms that they pulled out of the study is the WWP2 gene, and WWP2 has been seen to be an oncogene, which downregulates other tumor suppressors. Part of my role in the biological sciences is to validate these results in a wet lab setting. To do this, I am using a series of cell lines known as the WPE1 series, which was developed by a research group at Michigan State University. This line was artificially developed by exposing an epithelial prostate cancer cell line to carcinogens to develop cell lines which represent each of the 4 main stages of prostate cancer progression. My role is to look at the expression of the novel isoforms in these different cell lines. And I do this by first extracting the RNA from the cells, converting it to a cDNA copy, and then running QRT-PCR to quantitate the amount of transcript present. My results show this isoform to be upregulated in stage 3 of prostate cancer progression, which is the early stages of metastasis and can lead to disease and death later on. As a result, I predict that it could serve as a biomarker to predict patients who are likely to advance from a non-harmful benign cancer to a more malignant form. As a result, this will help us determine which treatment options would be best, and it may help us to develop more effective and targeted therapies by learning more about its mechanism of action. Also, by verifying the results of the computer algorithms, this indicates that they could potentially be used in other forms of cancer to detect biomarkers for other various types.